Hello everyone. Today we are going to learn about a very interesting topic in genetic engineering, that is the plasmid vector pbr322. In today's lecture, I am going to explain about the overall view of pbr322, its structure, construction, how it is used in gene cloning process. So what is pbr322? It is an artificial human-made vector. It is one of the most widely used vector in gene cloning. The size of pbr322 is 4363. The packaging size of pbr322 is 10 kb, which means the maximum gene of interest that we can insert into the pbr322 is only 10 kb. So if our gene of interest is more than 10 kb, then we should go for the plasmid vectors. The number of restriction sites that are present in PBR322 are 40. Even though there are 40 restriction enzymes, only 29 are useful and remaining 11 are not functional because these 11 are present near origin of replication site or near the marker sites, and we can see there are two marker sites present in PBR322, that is the amphenolin resistant gene marker site and tetracycline resistant gene marker sites. These marker sites help us giving our gene of interest in the final outcome. They also help us in differentiating between the recombinants and non-recombinants. The main reason why PBR322 is widely popular because it has two marker sites, that is amphenolin and tetracycline marker sites. Due to this nature, it is very widely popular and which helps us providing our desired gene of interest at end of the cloning process. Let's let's talk about the nomenclature of PBR322. It is first developed by Bolivar and Rodriguez in 1977 in the laboratory of Herbert Boyer at the University of California, San Francisco. The P in PBR322 indicates the plasmid, whereas BR indicates the Bolivar and Rodriguez, the scientist who created this vector, whereas 322 indicates its number in PBR series. Let's talk about the construction of PBR322, how Bolivar and Rodriguez constructed the PBR322. It is mainly obtained from three sources. As we can see, the PBR322 has amphenolin gene and tetracycline gene and origin of replication site. The amphenolin gene is originally present on the plasmid RSF2124 of E. coli. The tetracycline gene is obtained from PSC101. It is also the species of an E. coli. The origin of replication is obtained from PMB1 plasmid coli1, which produces the colicin. So, Bolivar and Rodriguez combined these three features and created a new vector called PBR322. Let's talk about the structure of PBR322. The PBR322 has 40 cloning sites. Origin of replication or ORI is the point where the replication of our vector starts in the host cell. ROP gene codes for the restrictor of the plasmid copy number. What is copy number? Means the capacity of a plasmid to produce clones in the host cell is called as copy number. The gene BLA encodes for the amphenolin resistant protein marker, whereas the gene tetra A encodes for the tetracycline resistant protein. As we know, the PBR322 has 40 restriction sites. 11 of these 40 restriction sites lies within the tetracycline gene. There are two sites for the restriction enzymes HIND3 and CLA1 within the promoter of the tetracycline gene. There are six key restriction sites inside the amphenolin gene. Amphenolin codes for the beta lactamase in the cloning process. The tetracycline gene consists of two restriction sites, that is BAMH1 and SAL1, whereas the amphenolin gene consists two restriction sites, that is called as PUV1 and PST1. Let's talk about how PBR322 is used in cloning process. As we can see in the picture, PBR322 has origin of replication site, amphenolin resistant gene site, and tetracycline resistant gene site. Now using restriction enzyme BAMH1, we are cutting BAMH1 restriction site and inserting our gene of interest using ligase. Now insert the vector into E. coli and culture it in amphencycline and tetracycline antibiotics media, and then place it in the amphencycline media, and those containing the plasmid will grow. The tetracycline gene lost its resistance as we inserted our gene of interest, which indicates that our cloning process is successful, and only amphencycline gene shows resistant to the antibiotics. That's how we can successfully make recombinants using pbr322. The main advantage of PBR322 is two selectable markers which allows the easy selection of recombinant DNA, that is tetracycline and amphencycline. The main disadvantage of PBR322 is the limitation of its size for the gene of interest, and it does not have very high copy number. So if we insert the vector into an host system, it cannot produce more plasmids, which may create the less chance of our desired recombinants. Thank you, everyone.